Welcome to Module 1.2. In this module, we'll be talking about genetic diversity and some of the ways that we kind of preserve the genetic diversity that currently exists. Previously we talked about, plant improvement, crop improvement. And so some of these programs, have kind of started in the 1900s as population started to increase and we started recognizing that we're going to need more food and more crops in order to feed our growing population. So this has really been around for a while, but it really started as like traditional plant breeding. Most of that plant breeding kind of came from, as I mentioned before, we would find plants that had characteristics that we would kind of try and keep them in, keep and make sure that we take the best of those crops. But something that they didn't have though was how to, transfer those characteristics to other crops. And so, the ability to select favorable characteristics over unfavorable characteristics, we were able to kind of take those desired traits from one crop or from one species and then kind of introduce that into another species and make sure that we got kind of the best of both worlds. And you may have heard me recall me saying this as hybrid vigor. Taking about a half a century back, where plant improvement really started was with Charles Darwin and these evolution concepts. And when we're talking about Darwin's evolution, this isn't really kind of like, did we evolve from that primordial ooze, or how did humans really get here? Did we descend from uh, apes or whatever? That's really not what this particular concept was about. What it really was that there are certain individuals that have traits that allow them to survive and adapt to changing environments and to be able to pass that on to future generations. There are some advantageous traits, but there is also some variation. So it wasn't necessarily that this was a perfect concept or a system. It wasn't that once this trait was introduced into an offspring that it continued on in future offsprings. And so that work kind of got uh, expounded upon by Gregor Mendel. And, many of us might remember from biology, Gregor Mendel with the peas and kind of going about how these cross these, big A, little a with big B, little b, and what the offspring of those generations looked like. And so, we kind of credit Gregor Mendel with the foundation of modern plant breeding, or at least the foundations of that, right? So he at least gave us some guidelines and something to think about and expound upon. So, we got the 1850s, 1860s, these two scientists really kind of got the world thinking about what is it with these genetics and how does it work. And genetics, we have a pretty good idea about it, but it's still not concrete. There's still a whole bunch that we just don't understand. And so Gregor Mendel really kind of, as I mentioned before, kind of worked on Darwin's, thought processes on variation and just how to move those traits around genetically. So he really came up with, hybrid crosses, back crosses, test crosses, and those gave us the idea of like how we can like verify or make sure that we actually have the trait was actually passed on and we can continue to see that. So when we're thinking about these traits that we're looking for in plants, there's kind of the fundamental plant breeding equation, if you will, where we have this phenotype, which is kind of what we see, when you see a plant that has a lot of corn or a particular stature, or it produces X amount of yield, that is the phenotype, or kind of like the physical, what we can see. And that is actually a function of genotype, which is the genetics crossed with the environment. So like we found that environment plays a role in that as well because of Darwin's discovery of variation, and then also this kind of survival of the fittest. And so in certain environments, certain plants might have characteristics or traits or might adapt their DNA a little bit in order to overcome some of these environmental stresses. So you're going to see this phenotype by equals genotype by environment a lot. Kind of this is the central dogma for me of what plant breeding really is. So how can we manipulate? When we get into module 2, we'll start talking about how do we manipulate the genotype. And so when we talk about crop improvement, many of us are thinking genetically modified organisms. There might be quantitative trait locus. We're going to talk about marker assisted selection, and that is going to be specifically trying to alter this genotype or the genome. And then how does that actually help us overcome the environment so that we have these crops that we can continue to grow? So we have these wild type relationships with these adaptive variations, and so plant breeders will want to try and get as many of those plant genetic resources as they can. And plant genetic resources are really just having a multitude of different crops or different plant species that have very different characteristics that we can try and like paint from. Think of it like when I took plant breeding, my major professor kind of spoke of it that plant breeding is like an art and a science. So there's some science to it, and that's going to be kind of like the genetics aspect of it too. But how do you paint the picture, or how do you take the specific colors of each of those plants? I'm trying to use like colors and traits. How do you take specific traits or colors and paint the picture of the environment that you're trying to overcome? So how do we find these crops that have specific pest resistance that we can then breed into our traditional crops? And so we have crops that we typically grow, our domesticated ones. And then if you remember me talking about the centers of origin, that's where we might kind of go back and look for genetic resources that might have come from one of those centers of origin so that we can improve our pest resistance. Because if that crop still grows in this center of origin and it has this pest resistance, we've removed that and kind of made that crop grow in an environment that it wasn't necessarily adapted to. That's kind of what domestication is. So we've taken it away from its quote unquote home and then kind of put it into a different environment and have tried to cultivate that. So some of the ways that we go about improving our crops in environments that they don't necessarily supposed to exist in, we'll use those wild type crops, those genetic resources from those centers of origin to improve some of these things. And so just kind of a workflow of how we go about, acquiring where do we acquire it from? So we're talking about going to those centers of origin. How do we conserve it? So, thinking like botanical gardens, or I'm thinking of some long-term study where we're going to make sure that we have enough of this material and we're going to consistently, as we see over here, consistently multiply it and then regenerate it so that we have enough of it. So we call this kind of like stockpiling or bulking. And so that's how we manage those. And I feel like this particular module is going to discuss a lot of these top, the acquisition, the conservation, and the management of that. The characterization, not so much. That's really more like if you were to go into plant breeding, or if you were to go into, plant genetic resources and that conservation type of thing, where we're trying to ensure that we keep those particular traits and not lose them, then you would go into the evaluation and the documentation. For us for me as an agronomist, or for kind of us as we're moving into the ag field and having to get these crops and grow them so that we can feed the world, this is probably going to be more like module 2, where we start talking about these different breeding principles and the new biotechnologies that we have. And then, finally, in module 3, we'll start talking about how we utilize some of these some of these crops in order to meet the challenges that we'll face. So what are those plant genetic resources? It's really the basic raw material that we'll kind of go back to and pull from and go, well, we know that this crop, when it was grown in this particular region, had these genetic resources. So I think about this also kind of like maybe like a fertility bank when I'm thinking of humans, and maybe you want these specific traits of a particular individual and you document that, and you know that, you know, maybe this particular sperm donor has X traits. So, you know, they know that they have blue eyes or blonde hair or they're tall or whatever the case may be. When I'm thinking about a plant bank, I'm also thinking about like a fertility bank where we're going to get a specific genetic resource that we know is going to almost breed true to type. And so it's really all of that genetic variability found within a particular plant species. So you have to think about how many plants we have, and then how many different species there are and the different traits within them. So a couple of terms that we're going to need to know is going to be a land race. And so these are going to be primitive crops that have been, you know, from a small isolated area that possess very high variation. So there is a lot of room to work with. There are a lot of different traits that we can get from this particular land race. So multiple traits that we can pull from. And then also think about this when we're talking about genetic resources, is that we don't want to kind of cross into the same plants. We kind of call this inbreeding depression, where if you continue to breed the same plants over and over and over again, eventually you kind of wash out all of that genetic variation and you basically just get stuck with the same old plant. You don't get any improvement. Next, we have these weedy races, and pretty much these are going to be the same thing as land races, but they'll possess more weedy traits. And so, you know, the ability to adapt in a niche or not need a lot of resources or be able to take some other resources. When I was reading up on this, it was really kind of like some of those weedy traits will be like tall and spindly, so that you're still getting the plant. It's able to survive with very minimal nutrient or environmental resources and yet still reproduce. So we have a land race, a weedy race, and then finally we have these wild species. And so there are some things that we can cross with other certain crop species. And so this is going to be kind of what we do if we have bred out most of this genetic variation and we can't get any more improvement on a plant. And so we will bring in a wild relative that can kind of maybe bolster or kind of bulk up some of these other traits so that we can then start to improve that crop. And we'll talk about this wild species as we talk into traditional plant breeding methods in Module 2. So we have a lot of different plant species that have been described. We have about 3,000 that we use for human use, and about 30 of those plant species meet the majority of our needs. So, you know, we have gone from, 24,000 different plant species to about 30 that we know are going to be the ones that we really, really need. And so we can pull from some of these other plant species that aren't necessarily adopted or kind of cultivated in larger quantities, that meet this 95% of our needs. There are plenty of materials that we can pull from in case if these 30 plant species start to lose some of their hybrid vigor or their genetic diversity. So these plant genetic resources kind of have one of these Venn diagrams where we have our land races, and these are kind of, different cultivars or breeding lines, maybe even some modern cultivars that we're using so that we can at least meet the needs of our current situation without like ruining our future situation. For some of these other plants, we have these plant genetic resources for like non-food utilization. We still need these ornamentals. They kind of give us a sense of pleasure and enjoyment, recreation. Maybe we're growing some of these crops that produce, maybe trees that produce the same amount of yield, I call it yield, in a shorter amount of time. And then ultimately, we are going to use these wild species to kind of increase or bolster or kind of bulk up some of these medicinal plants and these land races in order to overcome our future challenges. So when we talk about genetic diversity, it sounds exactly like what you think it is, right? It is all of that genetic information that is contained within a plant species, a population, or group of individuals, or whatever it is, right? So you have to think about all the possible genetic combinations that exist within a particular family, like a plant family. Like for instance, let's say the Poaceae family. Yeah, it's mostly all of grasses. Think about how many different grasses that we have. And then I want you to think about how each of those different grasses have different traits. So this genetic diversity, we have plenty of it. What we need to make sure is that we're preserving that genetic diversity. And so this is really going to be, like I mentioned, a lot of those different genes and those traits that are present within that given population. And then also within that genetic diversity, what we want to do is we want to document or kind of characterize or evaluate what is going to be that variability, and then how can we use that variability to our advantage down the road so that we have this long-term survival, again, that we want to make sure we maintain this particular genetic diversity in case if in like 25 or 30 years that we don't have that genetic diversity anymore. We've lost that variation and that adaptability. Obviously, this is going to be important, right? Because we're going to need more resilient crops. We're going to have that changing environmental conditions. We also need to have like a large genetic pool for this trait selection and improvement, because if you're only pulling from, there's only so many different combinations that you can have. If you only had six particular, 6 plants or 6 species that you could pull from, like at some point in time, you wouldn't be able to cross those anymore. And so as you can see, this is going to reduce our vulnerability to most of these stresses that we're going to be facing soon. And we can also create new crop varieties with improved agronomic traits. So, we want to, this is where we get back to those more edible parts or larger edible parts, or we have like herbicide tolerance or pest tolerance. Those are the types of things that we need this genetic diversity for, and to have that characterized. So we want to get, I think these next couple of slides are going to be a lot of definitions and just kind of get some terms down. And you'll probably see these terms several times because they're kind of the foundation of what genetic diversity is. All right, so we have this allele, and this allele is going to be this gene. There are alternate forms of this gene, and those occur within a specific position on a chromosome. So like in module 1.3, we'll start talking about meiosis, and we'll kind of go back over some of that biology stuff to kind of reinforce what we're actually doing and why traditional plant breeding methods can be so difficult, and why we're looking to biotechnology in order to improve that. So you'll hear us talk about different alleles a lot. Next, we have the gene pool. Many of us have used this term kind of within the human population, but we also have this as well in our plant populations. And so that's really just the total collection of these different alleles within a population. So this is like the genetic diversity of this particular group. So we want to have a lot of options and a lot of different traits that we can cross so that we don't really kind of limit ourselves in what we can and cannot do within crop improvement. Next, we have a mutation. And so there are kind of three traditional plant breeding methods that we use. One is mass selection, one is going to be hybridization, and then the other is going to be mutation. And so over time, certain sequences of genes on the chromosome have kind of been altered and have caused some genetic variation or produced some new positive or even negative trait. And so that is one way. That is kind of the way that we found these genetic variations. If we go all the way back to like Darwin and Mendel, if it was something that just stuck out, we're like, I've never seen that before. I wonder what happened. In the 1850s, they didn't know about DNA and chromosomes, but it is these genetic these mutations that give us also new genetic variations that we weren't really expecting. Genetic variation. It's just going to be the diversity of those genes and alleles. So you can kind of see that these alleles are part of this gene pool as a collective whole. We have mutations of those alleles that give us this genetic variation. And in the next, we'll talk about gene flow, and you've heard me talk about genetic recombination. All right, so gene flow is just the movement of that genetic material between different populations of the same species. And so we're not able to cross, corn with cotton or corn with soybean. So they have to kind of be in the same species, and there are certain frameworks that we can and cannot bring some of these things with. So gene flow will also kind of go into our genetically modified organisms, as I mentioned in the golden rice, where they took the daffodil and they took that bacterium and they took some of that DNA that worked with rice in order to produce this vitamin A. Genetic recombination, just what you think it's going to be. So in meiosis, there's going to be this, what we call crossing over. And when those two chromosomes cross over, that's where we get that genetic recombination, or at least the potential for the genetic recombination. And so this is when we start getting into DNA. We'll discuss that a little bit, but I don't really think that this course should be too focused on those molecular genetics as it previously was. And so when we talk about this in meiosis, we're gonna talk about gametes. All right, so now we have genetic drift. And so if we get a plant species, that gets the way that a seed might move is, maybe through wind movement, or it gets carried by an animal, or maybe a bird eats a seed and then flies to a different location and then drops that seed. There won't be any other compatible species for that to recombine with, or to continue on, or create progeny. So there's going to be this fluctuation of these allele frequencies, or how often this particular trait occurs within the next population. And if we start losing certain of these alleles, then we start reducing this genetic diversity. And so this is kind of what we are experiencing, or will run into. And when we think about plant breeding and our traditional methods, we don't have many more of these alleles left. So we're going to be looking to those centers of origin to bring in different species so that we can improve or bring back some of this genetic diversity to help us in the future. We had this founder effect. So this is also what I was talking about, when you have that seed that transfers into a new location, and there are only a small group of these individuals that are genetically distinct from anything else. And so this will be different. That genetic diversity, because of that limited gene pool, a limited gene pool equals limited genetic diversity. And so then eventually it's possible that when you take out all of that genetic diversity of this particular little small group of individuals or population, eventually they'll die out. And so then we lose that particular, those genetic resources inside of that gene pool. A bottleneck effect, this kind of goes along with that founder effect in that, we lose some of that genetic diversity because there's no other plant material or genetic variation in order to continue on those lines, right? So we get this dramatic reduction in that size of that population. And if we don't have many other plants to combine with, then we lose that genetic diversity. And ultimately, we'll lose that whole population. So we got the founder effect, which is kind of like, when we get a new population that's kind of different, and because it's different, it might not be able to combine with or be fertilized and then reproduce because the genetics are so different that they're just incompatible, which will kind of run us into that bottleneck effect. And so here we have the selective pressure. And so this is going to be kind of where we talked about with Charles Darwin, that survival of the fittest, and that's going to be kind of dictated by environment. So when I think about this, consider if you had a forest and there was an animal that dropped a seed. And the seed is able to grow for a little bit, but eventually that the forest canopy blocks out that light. And so that plant either learns to adapt to that low light environment or it will die. And once it adapts to that low light environment, well, then we now have this new plant population or this new founder that has these specific traits that allows it to grow in a not so ideal environment that it probably previously came from. And so that is going to lead to these adaptive changes in that population. Okay, so next what we're going to talk about is this term kind of germplasm. And so, like Nikolai Vavilov was kind of going across these different regions and he was recognizing that there were different plants and he wanted to take those back, and we want to use that germplasm or that genetic variation to introduce into existing crops that can become new crops. So you might hear the term germ, and all that is just the entire collection of genetic resources for that organism. Next, we have germplasm. And so germplasm is actually the living tissue from which those new plants can be grown. And that living tissue might be a leaf, or it might be a seed, or it might be pollen. It's going to be something that we can, something that is viable, that we can then either, bulk up, or maybe we have a lot of seed that we can kind of keep in maybe a tube. And we know that we have enough of those seeds. If we grow one plant, we can take one seed out, or maybe 2 or 3 seeds, and start to kind of grow a population so that we can continue to bulk that. So we would look for our germplasm. As I mentioned before, this is going to be maybe a seed or a leaf or a stem, pollen. Sometimes it could even just be a few cells that we can reproduce with, as long as they have, those units of heredity. So when we talk about units of heredity, we're looking at those chromosomes and those genes that we can then pull something out of.